Okay, so today we're going to be talking about the drug acetazolamide. So first we'll be talking about the cause of the disease, then its nature and its mechanism, the side effects, and then discoveries. So onto the cause. So acetazolamide, also known as Diamox, is used for swelling, which is also known as edema, heart diseases, seizures, glaucoma, and altitude sickness. And acetazolamide is also a diuretic drug as it causes your body to make more urine and lose excess salt and water to mitigate the swelling. And more into the specifics of the causes. So glaucoma damages the optic nerves, and secondary glaucoma is also present where there's a buildup of pressure in the eye. And for edema, there's swelling and buildup of fluid, which is due to medical side effects and heart failure.
Okay. Acetazolamide is also used to treat seizures, some of which are caused by cerebral edema, and altitude sickness, which has symptoms including fluid buildup in the lungs and/or brain. The common thread of most of the diseases that acetazolamide is used to treat has to do with with swelling or edema. On the cellular level, this edema is caused by a failure of sodium pumps in the cell membrane that then cause an imbalance of ion concentrations within the cell. This makes the cytosol of the cell hypertonic to its outside environment. Then the cell then compensates by drawing water into itself, causing the cell to swell.
Okay, now we're going to talk about the nature and mechanism of acetazolamide. So how it works is that acetazolamide is a non-competitive inhibitor of the enzyme carbanoic anhydrase, which prevents the breakdown of carbonic acid, causing an excessive accumulation of this acid. So in turn, this will end up lowering your blood pH, as well as regulating the activity of ligand-gated ion channels, such as GABA-A signaling, which slows abnormal electrical activity, which prevents seizures. The inhibition causes sodium bicarbonate and chloride ions to be secreted instead of absorbed, which leads to the excretion of water to maintain equilibrium. This decreases the blood pressure as well as reducing secretion of the aqueous humor, which lowers the intracranial and intraocular pressure.
Next, we're going to look at the mechanism of how this drug works. It begins with the exchange of sodium ions and hydrogen ions across the cell membrane. The hydrogen ion is going to form carbonic acid with the bicarbonate ion. The carbonic acid then decomposes into carbon dioxide and water. All of this is catalyzed by the enzyme carbonic anhydrase type 4. Then the carbon dioxide and water that were just made are going to be secreted into the cell, where they are going to react again to form carbonic acid, which is also catalyzed by a second carbonic anhydrase enzyme 2. The carbonic acid that was just made is then going to be converted to a bicarbonate ion. That ion and sodium ion will then transfuse into the bloodstream from the cell. Acetazolamide is going to inhibit both carbonic anhydrase enzymes in the lumen and the cytoplasm. The enzyme catalyzes the reaction of carbonic acid to bicarbonate ions in both places. And when it is not working, this is going to decrease a lot of the bicarbonate ions and prevent them from going from the cell into the blood. Because of this, bicarbonate ions and sodium ions will stay in the lumen and be excreted with other waste products instead of being absorbed into the bloodstream.
Okay, and then next I'm going to be talking about the side effects of acetazolamide. So there are many adverse effects. The main four would be high acid levels, liver injury, Stevenson-Johnson syndrome, and then white blood cells. So high acid levels, also known as metabolic acidosis, is a condition where there's too much acid inside the blood. It can be caused by a buildup of carbonic acid and can result in kidney stones, brittle bones, or even slow growth in children. Liver injury, also called hepatotoxicity, is the mechanism involving hypersensitivity related to its sulfonamide structure. It triggers an immune response to liver cell damage. Stevenson-Johnson syndrome can be caused by genetic background, but it's an allergic reaction causing the immune system to attack the skin itself. This can cause blisters and different peeling of the skin. It can also affect the mucous membrane. White blood cells, mainly leukopenia, but there's also many other blood cells or blood disorders that are affected, such as aplastic, anemia, and hemolytic anemia. But this causes a destroying of the white blood cells, which are vital to help the infection of it, fighting infection. So each of the main adverse effects also has different effects of its own, such as high acid levels. It can cause confusion, weakness, tired feeling, headaches, and then fast or irregular heartbeats. Liver injury can cause nausea or vomiting, stomach pain, fever, tiredness, itching, or even a loss of appetite. Stevenson-Johnson syndrome can cause a painful red or purple skin, rash or blistering around the mouth, skin, or nose, or red, painful, watery eyes. And then white blood cells can cause fever, shortness of breath, yellow skin, headache, or also an abnormal heartbeat.
Finally, we will talk about the discoveries that led to the development of the drug. So there were three major discoveries that led to the discovery of acetazolamide. And the first is the discovery of carbonic anhydrase. So this was discovered in the 1930s, and it's an enzyme that catalyzes the reaction between carbon dioxide and water to create bicarbonate and protons. Now, this was found at the time to play a pivotal role in pH stability in the renal system, and therefore a lot of the other developments based around that. And following a decade after, in the 1940s, sulfonamide was developed as an antibiotic agent. However, one of the side effects was it also was found to have an inhibitory effect on the enzyme carbonic anhydrase. Finally, a decade after that, in the 1950s, a team of scientists decided to put the two developments together and synthesize various derivatives of sulfonamide in order to inhibit the enzyme. This discovery was specifically driven by Richard Owen Roblin Jr. in the 1950s and his team. This led to the actual development of the drug, and he and his team took the first compound found in the top right, which is a key functional group of many sulfonamide drugs, and synthesized it by adding different branches and allowing for the thiodiazole ring to bind to the enzyme and to become an inhibitor to the carbonic anhydrase. And since the carbonic anhydrase was found in the renal system originally, it was originally designed to induce diuresis or to allow the body to excrete even more urine. However, as the enzyme was later found to play a pivotal role in red blood cells, the gastric mucus, and the pancreatic cells, it was also expanded for a variety of medical conditions that was found and used today. And these are our works cited pages over here. Thank you for watching.